Hello students, welcome to EPG Pathshala. I am Dr. Shormishta De from Department of Biophysics, All India Institute of Medical Science, New Delhi. Today I am going to talk about peptide synthesis, definition, methodology and application from pepper techniques in molecular biophysics 1. Peptides are present in the living cell as enzyme, hormone, antibiotics and receptor and have diverse biological activity. The main objective of peptide synthesis are to study the relationship between structure function of biologically active protein and peptides and establish their molecular mechanism. To synthesize peptide, those are clinically important such as antibiotic, vaccines and other drugs to develop new peptide based kits for immunogen. Peptide is the amide-like linkage between two amino acids. Peptides are the prime important in the regulation and maintenance of all biological process. The variation in properties of the protein depends on the nature of the side change of amino acid. In amino acid, they have a different side chains like hydrophobic amino acid, acidic, basic. So the properties of the protein depends on the side chains of amino acid. Types of amino acid: hydrophobic amino acid, phenylalanine, glycine, alanine, isoleucine, leucine, valine. Side chains are nonpolar. They are water-hating group and usually located interior of the protein. Hydrophobic interaction is the important aspect for the stability of protein. Stabilize the folded conformation. Glycine found in the turn region of the protein where the backbone has to make sharp turn. Basic amino acid: arginine, histidine, lysine. They are positively charged at physiological pH, involved in electrostatic interaction with negatively charged group of amino acid. They occur in the active side of the protein as it can function as very efficient general acid-based catalyst. Negatively charged amino acids are aspartic acid, glutamic acid. They are negative at the physiological pH. They are usually found on the surface of the protein where they can interact with favorable solvent molecules. They take part in the electrostatic interaction with positively charged amino acid. The amino acid serine and threonine are not very chemically reactive. Tyrosine and tryptophan amino acids found buried in the hydrophobic interior of the proteins allow hydrogen bonding interaction with other residue or even with solvent molecules due to the presence of hydroxyl and amino group in their side chain. Asparagine and glutamine amino acids are having a amide group in their side chain, which are usually hydrogen bonded whenever they occur in the interior of the protein. Proline amino acid are very rigid amino acid. Cysteine and methionine amino acid form a disulfide bridge involve SS bond. Cysteine frequently occur at the metal binding site as the sulfur atoms can form covalent bond with the central metal ion. Why peptides are important? Because of restriction of conformational flexibility will lead to high degree of predictability. In the peptide bond, that is CONH bond, the rotation is restricted. So the restriction of conformation flexibility increases the predictability of the conformation, and so they are highly active and so small amount is required for biological function, because the small amount which act very rapidly, and they are highly specific and have therefore relatively low systematic toxicity. As they are highly specific and highly reactive, so the toxicity does not develop easily. It do not accumulate in the body or for the short life. It metabolize very soon after the reaction. They are easily acceptable by the body with very slow side effect, because they are not toxic and specific and do not accumulate. That's why the side effect is very low. The amino acid react with the carboxylic of next amino acid to form a peptide bond. This polymerization of amino acid creates a protein. So, when two amino acids react with each other, the amino group of first amino acid react with the carboxylic group of another amino acid to form a peptide bond CONH and forms a dipeptide and release one molecule of water. Conformation of peptide. The conformation of peptide obtained by the rotation around a single bond, that is C alpha and N and C prime and C alpha. The rotation around the bond C alpha and N and C alpha and C prime are given by a dihedral or torsion angle, that is phi and chi respectively, and the angle omega is 0 or 180 due to the delocalization of carbonyl pi electron and nitrogen lone pair between C prime and nitrogen. The key to protein folding mainly lies on the torsion angle phi and chi of the backbone of the amino acid. The important feature of peptide link are its planarity and short length between C prime and N. The bond length between C prime and N is 1.32 angstrom, which is usually 1.47 angstrom, and the bond length between C prime and O is 1.24 angstrom, which is usually 1.43 angstrom. The peptide bond NH and CO is planar and always in trans configuration. C alpha and N and C alpha C prime are always trans to each other with respect to the peptide bond C prime N. As the rotation around C prime N is restricted and the flexibility of the polypeptide chain is only due to the rotation around C prime and C alpha and N. The shortening of the bond length is due to the delocalization of the double bond of the carbonyl group is in between C prime O and C prime N. This resonance of the 6 atoms tend to lie them in a plane. The shortness of the bond and the partial double bond character gives more strength, and rotation around the bond is restricted, which give rise to planar structure. There are two methodologies of peptide synthesis: solution phase peptide synthesis, solid phase peptide synthesis. In case of solution phase peptide synthesis, the synthesis takes place in the solution throughout the reaction, and after every coupling, the peptide is isolated and purified. In solid phase synthesis, a solid support, that is resin, is required, which is attached to the carboxylic group of the amino acid, and after the synthesis, this resin is cleaved to get the free peptide at the carboxylic group. Amino and carboxylic groups are required to be protected by protecting group, as in peptide synthesis, two amino acids are involved to form a peptide bond, and both the amino acids contain a carboxylic and amino group. So the carboxylic group, one amino acid, and the amino group of another amino acid should be protected. So there are different groups are required to protect this amino group and carboxylic group. So here we are showing the protected group of amino acid, BOC, that is tertiary butyl carbonyl, which is blocked the amino group of amino acid. For that, tertiary butyl oxycarbonyl chloride react with the amino group in presence of sodium hydroxide to protect the amino group by BOC. Benzyl oxycarbonyl chloride is react with an amino acid in presence of sodium hydroxide to protect the amino group of amino acid by benzyl oxycarbonyl. Another group which protect the amino group of amino acid is FMOC, that is fluoroenyl methyl oxycarbonyl. Amino acid and 9-fluoroenyl methyl chloroformate in dioxane are stirred overnight to protect the amino group by FMOC. Another one is acetyl group, which is deacetylated and block the amino group of amino acid by acetyl group. Now, the carboxylic group protected molecules are methyl and ethyl ester, which are stirred 24 hours by methanol or ethanol to protect the acetyl group by methyl ester and ethyl ester respectively. After the synthesis of the peptide, these protected groups are required to be removed. So deprotection of amino group. BOC group is removed by tetrahydrofuran used for deprotection of BOC protected peptide. This group is deprotected by reacting tetrahydrofuran with DMF and stirred for 1 hour to remove that BOC protected group. Now FMOC is removed by piperidine, which is also stirred for 1 hour to remove the FMOC group from the peptide. Now acetyl removal is done by potassium hydroxide to remove the acetyl protected peptide. Activation of C-terminal carboxylic acid. Before starting the coupling of two amino acids, one amino acid is need to be activated at C-terminal part of the amino acid. These are the molecules which are used for the activation of C-terminal of amino acid. Carbodiamide such as dicyclohexyl carbodiamide or diisopropyl carbodiamide, N-methylmorpholine, HOBT, hydroxybenzotriazole, HBTU, 2,1-H, benzotriazole, 1-IL, 1,1,3,3, tetramethyluranium hexafluorophosphate. Different mechanism of amino acid coupling. There are different mechanisms to couple the two amino acids. These are the mechanism: mixed anhydride coupling, DCCI-HOBT coupling, salt coupling, acyl halide and pseudo halide coupling. In mixed anhydride coupling, NHS and EDC are used for the coupling method, and in DCCI-HOBT coupling, it activates the carboxylic group by DCCI and HOBT, and in salt coupling, NaCl are usually used for the salt, and in acyl halide and pseudo halide coupling, the halogens are used for the coupling. But in among these four coupling method, the first two, that is mixed anhydride coupling and DCCI-HOBT coupling, are used more in the practice for peptide synthesis. Mechanism of peptide synthesis. Mixed anhydride coupling. In this method, BOC protected amino acid react with N-methylmorpholine which is the activator which activate this Boc protected amino acid by removing hydrogen atom from carboxylic group to make it negatively charged, which react with isobutyl chloroformate, and then it forms an intermediate compound with this isobutyl chloroformate and N-methyl morpholine hydrochlorides removed from the reaction. Now this intermediate compound, which is formed by reacting with isobutyl chloroformate, react with another amino acid where N-terminal is free and carboxylic group is protected by methyl ester. This coupling takes place at 0 degrees centigrade for 2 hours, and then it forms a peptide and leaves the other compound showing in the figure. The another mechanism of peptide synthesis is carbodiamide coupling. In this method, the Boc protected, which is protecting the amino group of one amino acid, react with dihexyl carbodiamide and activate this carboxylic group of the amino acid and forms this intermediate compound, which then react with an HOBT to make the carboxylic group reactive and forms an intermediate compound and release deoxycarbodiamide urea, and then this intermediate active compound react with the second amino acid, which is protected by methyl ester at the carboxylic group, and this all coupling takes place at 0 degrees centigrade to form another intermediate compound, and which then after 2 hours forms a peptide, that is a dipeptide. Usually in the peptide synthesis, this mixed anhydride coupling and carbodiamide couplings are generally used, but there are other mechanism also for peptide synthesis. These are acyl halide and pseudohalide coupling. In acetyl chloride, the activation of acetyl amino acid by conversion to the corresponding acyl halide followed by the reaction with amino acid in organic solvent. So acetyl glycine in presence of thionyl chloride and PCl5 form an acetyl glycine chloride, which then coupled with another glycine which is protected by methyl ester at carboxylic group in presence of magnesium oxide form a dipeptide acetyl glycine OCH3. In acetyl cyanide method, protected amino acid bromide can be converted to cyanide, which react with an amino ester to give a peptide. So the acetyl glycine bromide react with glycine OCH3, that is the another amino acid protected by a methyl ester at carboxylic group in presence of hydrogen cyanide to form a dipeptide, that is acetyl glycine OCH3. Acetylazide, protected amino acid converted to hydrazine and then react with amino acid ester to form a peptide. So amino acid which is protected at both sides with methyl ester at carboxylic group and is react with an hydrazine to forms an hydrazine amino acid, which then coupled with another amino acid where again the carboxylic group is protected by methyl ester and presence of a nitrous acid to form a dipeptide. This is the scheme which showing the solid phase peptide synthesis. In solid phase peptide synthesis, one amino acid is blocked by resin at C-terminal part, which is the solid support, and the amino group is blocked by FMOC amino acid. So, the first, before coupling with another amino acid, the amino group is deblocked, that is blocked by FMOC by pyridine, and then the other amino acid where the carboxylic group is free and amino group is blocked by FMOC. So this amino acid is activated by an activator N-methylmorpholin and HOBT to activate the carboxylic side of this amino acid. So then this amino acid, which is activated at carboxylic part, and other amino acid where the C terminal is blocked by a resin and the N terminal is free, is coupled, and to form an amino acid peptide where the carboxylic group is attached to the resin and the other part of the amino acid where amino group is blocked by FMOC. So this dipeptide is finally deblocked this amino group by removing FMOC by piperidine to make the amino group free, and this amino group can react with another amino acid and can increase the peptide chain from this N-terminal position. And to make the peptide free from the resin, finally the resin is deblocked by different method to get a peptide which is free from both the group. In solid phase peptide synthesis method, to monitor the completion of the reaction, the Kaiser test is done. So in Kaiser test, the presence of free amino acids are detected. Three solutions are required for this test. Solution A, that is ninhydrin in ethanol. Solution B, that is phenol in ethanol. Solution C, that is potassium cyanide in pyridine. Free amino groups gives blue color bits. So when the peptide is formed between two amino acids, the amino groups of one amino acid is blocked by FMOC group in solid phase, and the carbonyl group, carboxyl group, is blocked by a resin. So the amino group is always blocked by FMOC or any other protected group in a peptide. So before cleavage of this amino group, the Kaiser test is done to see whether free amino group is free or not. If free amino groups gives a blue colored bits which shows the incomplete coupling, and white colored bits shows the presence of free amino group. Now the cleavage of the resin. After the completion of full chain of peptide, to free the carboxylic group of one N, which is protected by a solid polymer, that is resin, the cleavage procedure of resin is done by washing the resin with DMF, that is dimethyl formamide, methanol and water successively. Then this resin is dried in a desiccator till it gets totally dried. Then this resin is starred with trifluoroacetic acid, water and scavenger. Scavengers are anisole or thianisole depending on the amino acid used for 2 hours, and remove it under reduced pressure. So all the solvent is removed from the resin by the reduced pressure. Then this ether is again dried in a cold condition to get the solid peptide. Remove this ether and add 10% of acetic acid, and after that it keep in the fridge to get solidify and then that is lyophilized to get the dry peptide. This is the procedure to cleave the Wang resin. Now the cleavage of the PAM resin. Almost the procedure is same, but the reagent which is used for the PAM resin is trifluoromethane sulfonic acid, TFMSA. The scavengers are used during the cleavage to protect the reactive sites of the peptide during extended cleavage reaction time. Protected from the carbocation, water is used to suppress the alkylation during the cleavage of resin. Difference between solution and solid phase peptide synthesis. In solid phase peptide synthesis, solid support at C-terminal end is required. In solution phase peptide synthesis, no solid support is required. C-terminal is protected with methyl or ethyl group. In solid phase, the synthesis is always proceed via N-terminal site. In solution phase, synthesis is possible from both N-terminal and C-terminal site. Solid phase synthesis is one-pot reaction. Solution phase synthesis, the isolation and purification is required after every step of coupling. Solid phase synthesis is very rapid method, but in solution phase it is very slow, labor intensive, as product has to isolate after every coupling manually. In solid phase the yield is very high, but in solution phase yield is low as it loses at every step of washing after coupling. This is the machine which showing in the picture for solid phase peptide synthesis by automatic peptide synthesizer. These are the reaction vessel. In this reaction vessels, the coupling of the amino acids takes place where the resins are stored for the amino acid coupling, and these three reactions can take place simultaneously in these three vessels. This is the nitrogen gas connector which is connected to the nitrogen gas cylinder. All the procedures takes place through this nitrogen gas. This is the bottle which contains the solvent, that is DMF, for the reaction. This is the bottle which contains D protector, that is piperidine to deprotect FMOC. This is the bottle for the activator which contains N-methylmorpholine, which activate the C-terminal of the amino acid before coupling. And this is LCD control panel where all the programs are monitored for the peptide synthesis, like washing, deprotecting, activating and coupling. This is the carousel rack. There are 45 zoles which contains amino acid vials for the reaction of peptide synthesis. Software loaded in the machine. 6 programs in the machine are to run each step of the synthesis as well as manual runs. The programs are built for start up the instrument, washing after every step of the reaction, deprotection, that is deprotecting the FMOC group of the amino acid by piperidine, activation, that is activating the amino acid, the C terminal of the amino acid by NMM and HBTU, and coupling, that is the coupling between the two amino acid to form a peptide bond. And the repetition of coupling means when the coupling is not occur totally, the coupling with more amino acids is required. That is another program which is called repetition of coupling. Why peptide can be used as an inhibitors? Peptides is less immunogenic. It easily absorbed through metabolism as are biomolecules. They are easy to synthesize. They can mimic protein-protein interaction, and synthesis of peptide is possible due to the stereochemistry of enzyme active site and charge density. So students, let now summarize what we have learned in this module. This peptide can be easily synthesized because two amino acids are linking between two carboxylic group and amino group of another amino acids are attaching and removing one water molecule to form a CONH bond, that is the peptide bond. It can be easily synthesized. It's less immunogenic because this peptide act very rapidly and do not form any side effect. It goes out of the body very easily. It easily absorbed through metabolism as any other biomolecules because these peptides are formed by the linkage of amino acid. So it is absorbed through easily by metabolism. Can mimic protein-protein interaction because the protein itself is the chain of amino acids, so it can mimic protein-protein interaction. High therapeutic index with low toxicity. The toxicity is very less because it acts very rapidly. It won't develop any resistance, so toxicity is very less because only amino acids are present in the peptide chains and no other chemicals, and it's chemically and biological, it has a chemical and biological diversity. So the peptide is very important molecule for therapeutic value.